Thank you very much for the invitation. I'm pleased to be here. I guess we've worked with a company and Indico and Gabor now for a few years, and it's been a pleasure. Most of this work has been actually done by a postdoc that I had in Brazil, from Brazil. She was working on this, and she was here a couple years ago to present the data. There's some additional data which I'll discuss today. So in terms of my outline for the next half an hour, I'm going to talk to you about the model system that we use for neurodegeneration and aging. We use little worms, C. elegans. I'll give you a very brief overview of DDW and aging. I'll talk about the model again and the results that we've obtained over the last couple of years. And I have, I think, one slide that will just give you some ideas in terms of future directions. So first of all, I have to give a pitch to the model system that we use. We've used the C. elegans now for about 8 years. And we think this is a wonderful model system because it's well more advanced and physiologically more complex, given the fact that it is an in vivo system compared to tissue culture cells, where one would use a very reductionist kind of approach, where you might lose a lot of barriers and various metabolic capacities. So the C. elegans, which is shown here in the middle, is sort of an in vitro system, but a living in vitro system, which is in the test tube. It's obviously not as complex as some of the model systems that we have. It's an invertebrate like fish, but obviously, as you'll see, in terms of the genetic composition of this model system, it's much more primitive compared to mammalian systems. But nevertheless, there's a great homology between many of the genes that are found in C. elegans and in humans. And therefore, we can ask a lot of questions about what's the role of different genes in mediating disease. Now, this model system has about 50 hours of life, egg to egg. It's very easy to culture. You can just put them in 20 to 25 degrees and get hundreds of thousands of worms. You can freeze them when you don't need them, so you don't need very much equipment, and it's very easy to get to the different lines that you might have. And of course, as I already mentioned, the power of this system is based on the genetics. You could do very easily screens for suppressors and complementation. And the system is also very primitive in the sense that it has only 959 cells. And of these, 302 are neurons, and there's only about 7,000 synapses. So for those of us that are interested in the nervous system, it's obviously a very, very primitive system, which is very easy to look at. And I'll show you actually one movie later on to convince you that we can hone on specific cell types and ask various questions about their sensitivity to various agents, and then see how we might be able to rescue them. There have been a couple actually Nobel laureates who've received the prize for working in C. elegans. Both the GFP Nobel Prize, which was awarded a couple of years ago, as well as the work that's been done on apoptosis, has all originated from C. elegans. And since it's been worked out so much, we know there's a complete cell lineage that has been characterized all the way from the zygote to the adult. Now, C. elegans has less genes than we do, of course. There's only about 19,000 genes. You could see that only about 5,000 of them are essential. But as I mentioned before, there's great homology between the genes in C. elegans and mammalian system. And this is important because if you're asking questions about the function of the genes, it is important that they have similar compositions to the one that are found in humans. And given the fact that they don't have all the genes, what you could do is you could take certain genes that are not expressed in C. elegans and see whether they can hasten or alleviate disease. This can be done within the context of Parkinson's disease, for example. C. elegans does not express alpha-synuclein, But if you take alpha-synuclein and you put it within C. elegans, they become a lot more sensitive to MPTP and 6-hydroxydopamine, which are agents that are known to cause symptoms that are similar to Parkinson's disease. Vice versa, you could take other genes that are not expressed in C. elegans. You can take something like Parkin. You can put it in C. elegans and then ask similar questions, and what you'll find out is actually that Parkin is protective against MPTP, 6-hydroxydopamine, and manganese. So you can ask many questions regarding the genetic modulation of disease using this species. There's a number of servers, and there's actually banks for different strains for those of you that might be interested. We don't have to generate all the strains that we might be interested in, because we could just call the bank and we pay about $7 and we get a new strain. So it's very, very cheap. The caveat here is that you have to be in the society. So you have to have some papers published so that you recognize somewhat of an expert in the area, and then if you call or if you write them an email to the consortium, which is found at the University of Minnesota, they will send you the different strains. And it's very common when we and others develop different strains that we deposit them in this consortium. So these are available pretty much at no charge. Now, what can we do in C. elegans? It's actually pretty amazing when you look at it. We can look at growth, we can look at movement, feeding behaviors. I'm not going to read everything. There's actually some studies now that show you could even look at behavior in C. elegans. There are some specific behaviors that they can be taught to do, which you can look at and determine whether an agent might have a significant effect on their behavior. Again, the strength of the system is that you could use the genetics, and you can look at the interaction between the environment and the genetic composition. Now, we're interested in Parkinson's disease, and I'm not going to say very much about it. But this is just to show you how the nerve, the scheme of a dopaminergic neuron, which is shown here within C. elegans and mammalian systems, it looks exactly the same. What you could see is the different names. Those of you that are biochemists probably know this, but if you have tyrosine, it's hydroxylated to form the dopa, which then forms dopamine, which is packaged within vesicles. When you get the action potential in the presynaptic neuron, you get release of these vesicles, and the dopamine acts postsynaptically on some receptors. Some of it is also transported back on what's known as the dopamine transporter, and it's recycled and repackaged again via the vesicular monoamine transporter, and then it's available again for release. I'm showing you this because in C. elegans, the whole machinery that we find in humans is also found in this worm. There are differences in names. So instead of tyrosine hydroxylase in the worm, the gene is called CAT2. Instead of DAT, the gene product is known as DAT1, and so forth. I'm showing you this because what we can do is we can take green fluorescent proteins and tag them to the different genes in this pathway or in any pathway that you might be interested in. And then we can view those GFP-labeled neurons or any other cell under the microscope. And because of the small number of cells within C. elegans, it's very easy to follow what happens to these cells. You can imagine in mammalian systems, in humans, we have tens of thousands, if not more, dopamine-containing neurons. In C. elegans, we have 8. Okay? So it's very easy to see what different agents do to these neurons under experimental conditions. So this is what we do. We label the neurons with GFP, putting it in front of the promoter for the dopamine transporter, which I've shown you in the previous slide. And then, can we turn the lights off? And then we can look at these worms. It's okay, maybe you could see it. So this is how it looks. So I don't know if you can see it in the back, but there are 6 neurons in the head. They're green because they have the GFP in front of the promoter for the dopamine transporter, and there's a couple of them in the tail, which you could see down here at different places. So now we can treat with manganese or mercury or any compound that you might have affection for, and we can see exactly what happens to these neurons, and then we can see also whether you can rescue them. So this is all I'm going to say about the model system. In terms of aging, C. elegans is a good model for aging as well. As in humans, the worms do experience age-related neurodegeneration, sarcopenia, and as shown here, they accumulate lipofuscin. So this is a day 5 adult, and this is an adult at about 16 days of age. One thing that I forgot to mention, actually, and I need to go back, is over here. Now, I talked to you about the number of cells, that the strains can be frozen, the lifespan, and so forth. This is the cycle of the animal at 25 degrees Celsius. And you can see you go from an egg to the first larval stage at about 8 hours, second larval stage, 7 hours, and so forth. The important thing is if you stress these animals, if you put them in cold, if you do not provide them with food, what they do is they go into what's known as the Dour Stage. And the Dour Stage is a condition where they can actually be, they can remain in the Dour Stage for months. If you then refeed them, they go directly into the L4 stage, so they skip the larval stage 3, and then they live for a few more hours, and they lay their eggs and they die. So you can imagine that there's a lot of interest in this Dour stage because it's essentially a stage where you prevent aging. The animals just stop aging. They can be maintained and frozen in this stage for months without anything happening. So in terms of D2O concentrations exceeding natural levels, resulting in adverse effects, I think not much has been mentioned. We talked about the other spectrum of effects, and I'll show that in the next slide. But it's been shown that there are increased numbers of viral mutations, or there's an increase in the mutation rate with high concentrations of D2O. Deuteration of synthetic estrogen hormones weakens their estrogenic properties. Deuterated enzymes exhibit conformational changes affecting their active sites. Along a temporal aging axis, the skin becomes enriched in deuterium. So older animals tend to have more, and D2O also reduces the lifespan in mice. This is the other spectrum of the effects, which is associated with depletion, and we've heard over the last couple of days there's many more references. These are the ones that I've been able to gather, and some of them are older, other than the Kong reference, which is listed at the bottom. But generally, in vitro depletion triggers cell death. It exerts influence on proto-oncogenes and tumor suppressor genes. It reduces the expression of genes that are induced by carcinogens, and as I mentioned, the references are below. Now, one of the pathways for aging is the DAF2, or the IGF insulin-like receptor pathway, which is included here. This pathway is found in C. elegans in all mammalian systems, and essentially you have an agonist that binds to this receptor. You go through a whole cascade where PIP2 is phosphorylated. You get increases in AK2, 1 and 2, activation or phosphorylation of DAF16. And you can see the DAF16 can exist actually in both conformational changes, one that's phosphorylated and one that's non-phosphorylated. Essentially, when it's non-phosphorylated, it's bound within the nucleus, and it causes transcription and increases the levels of various proteins that are associated with protection. When it's phosphorylated, it tends to be in the cytosol, and this pathway then is abrogated. You get less production of enzymes or proteins that are associated with mitigating oxidative stress. Now, the model that we use is specifically for this study is associated with manganese-induced toxicity. I'm not going to say very much about it. Manganese causes symptoms that are very similar to Parkinson's disease in humans. It's associated with mitochondrial dysfunction, energy depletion, and oxidative stress. So you can see the relationship to the DAF16 pathway, which I've shown you in the previous slide. And it activates both necrotic and apoptotic pathways. And the hallmark of manganese neurotoxicity is that it does cause neurodegeneration, specifically within dopaminergic neurons, which are the GFP neurons that I've shown you before in the movie. Manganese-induced toxicity can be recapitulated in C. elegans. This is what you see at the level of the worm. You see changes in the excretory canal. There are changes in other kinds of cells and in the epidermis. There's retardation of growth. These are L2 larvae. These are L1 larvae that have been treated with manganese. And we and others have shown that manganese readily accumulates within the worms, and actually the concentrations within the worms are identical to those that you find in mammalian systems. And one of the effects that I'll show you is also that manganese decreases the longevity of the worms. Now, the effect is dopaminergic. What you're seeing here is various strains with either GFP or red fluorescent protein, RFP. These are dopaminergic neurons. The PUNC47 are GABAergic neurons, cholinergic neurons, and chemosensory neurons. These are control animals, and these are animals that have been treated with manganese. And I should point out that there are two worms here and here, so you're comparing 1 to 2 and 1 to 2. Essentially, I don't know how well you could see it, but the only neurotransmitter system that's being affected by manganese is the dopaminergic system. So these are controls, and you can see that in the manganese-treated animals, there's complete disappearance of these projections, the axons, and actually if you treat with higher levels of manganese, you lose fluorescence even in the cell bodies. So this is a very sensitive model for manganese-induced toxicity, which we have used in the studies that I'm about to describe. So given the fact, as we heard over the last few days, that there are beneficial effects to DDW in a number of animal models, we hypothesized that DDW would have anti-aging effects in the C. elegans. In order to determine which steps in this pathway that I've described, the DAF16, we looked specifically at what steps are being modulated by manganese itself, and then we aim to discern whether these effects can be reversed or can be negatively impacted by the addition of DDW. And specifically, what we've looked at is the DAF2. We've looked at the expression of DAF16. We've looked at AK2, or total AKT, I'm sorry. And we've looked at one of the byproducts of the activation of DAF16, which is manganese superoxide dismutase, which is an antioxidant enzyme. So for the treatment paradigm, what we do is we take a synchronous L1 larva at the 1 stage of C. elegans. We expose them acutely to manganese for 30 minutes, and then we treat them with DDW for additional 48 hours. So the results are going to be shown in the next few slides. The first thing that we did was to treat C. elegans just with DDW to see whether that by itself would have an effect. And as you can see, in normal wild-type animals, DDW by itself actually has no effect. So this is the % survival over days. We treat 48 hours. Then we follow the animals until day 15, and essentially these curves overlap one with the other. The next thing that we did was to look at what happens under conditions where the C. elegans are actually exposed to manganese. So we still have survival, we have the days, we have what we call the control groups, which is 150 parts per million, which is in black. We have the manganese group with normal control conditions, And you could see that there is a significant reduction, not so much in the number of days that they live, but you could see that the number of surviving animal between day 7 or 8 and 15 is significantly reduced compared to the wild-type animals, which are shown here. And when we treat the animals with manganese, with either 120 parts per million or 90 parts per million of DDW, we can reverse this effect. So the animals essentially overlap with the animals that are not exposed to manganese. We've looked at the pathway, as I mentioned before, specifically at DAF16. And DAF16 is shown here. These are Western blots, so we're looking at protein levels. This is DAF16 corrected for a housekeeping gene, which is in this case is beta-actin. These are 150 parts per million, 120 parts per million, and 90 parts per million of DDW. So this is consistent actually with the fact we don't see any change, and it's consistent with the fact that we didn't see any change in longevity when we treated the animals in the absence of manganese. But when we treat the animals with manganese, consistent with what I've shown you in the previous slide, we see a significant decrease in DAF16 expression, and this can be rescued in a dose-dependent manner, both by 120 and 90 parts per million of DDW. We've looked at manganese superoxide dismutase, which is downstream to the activation of antioxidant response elements that are found within the nucleus, as I mentioned before. So this is one of the byproducts of the phosphorylation of DAF16. And again, if you just treat with DDW, there are no discernible effects compared to the controls. If you treat under control conditions or control water with manganese, you see a reduction in the production of manganese SOD, but this effect, again, dose-dependently, is reversed in the presence of 120 and 90 parts per million of DDW. AKT protein levels in wild-type C. elegans show the same type of pattern. No effect with DDW, a significant increase in the presence of manganese, and here we found actually only the 90 parts per million to be effective in attenuating this increase to levels that are still significantly different from the control, but nevertheless they're also significantly different from the manganese plus 150 parts per million of water. Now we also decided to see whether there's any significant effect of doing similar types of studies in C. elegans where there's loss of function mutation in the DAF16 itself. So this is what I've shown you before. These are the animals, the wild types in black. These are wild type animals. If you treat them with manganese, you see a reduction in the number of surviving animals between 8 and 15 days, but no effect on longevity. And then you can rescue this effect, as I mentioned before, with 120 and 90 parts per million. The first thing that you could see is that when you use the MGDF50s, which are animals with a loss of function mutation in DAF16, is that they're actually much more significantly affected compared to the wild types. So that says something about DAF16 actually being very important in modulating this effect. The other thing that you could see is that in these knockouts, the rescue is only partial. So if you use 120 and 90 parts per million of DDW, you cannot bring these animals back to the control levels. And that obviously means that other pathways other than the DAF16 are probably involved in this effect. And what they are, I think we have some idea about what they might be, but we have not studied them. And finally, the next couple slides will show the relationship between survival, DDW, and loss of function or mutation in a couple of the apoptotic genes. These are the SED3 and double knockout SED3, SED9. So again, just for orientation, survival and days, 100 parts per million. N2 is the wild type worm. This is what you see. With manganese, you see that there is a significant effect on the number of days these animals live. If you use animals with loss of function mutation or complete knockout, actually, of SED3, you don't see any effect of manganese, which suggests that the effects of manganese are probably mediated by apoptotic pathways. And you see the same thing with SED3, SED9 double knockout. So this shows, when you compare these data to what you see with manganese alone, that the effect of manganese is mediated by apoptotic pathways, and if you knock out those genes, actually you don't see any effect. Now, we tried also to rescue these animals in the presence of DDW 120 and 90. Not surprisingly, we don't see any effect, because if you don't have the gene, you don't see any effect. So I decided to include it because it's, in a sense, negative data that are important. So this is 150 parts per million in a knockout of SED3. You don't see any effect with the manganese, so there's no rescue. And you see the same thing with the SED3, SED9. Again, the same kind of effect. So the conclusions are that DDW has anti-aging activity, not so much in the sense that it makes the animals live longer, but it seems to be able to rescue the animals so that more animals live at a given time period, which would translate also probably to a better quality of life in C. elegans, if you could measure that. We think that this activity is partially mediated by modulation of the DAF16 pathway. As I said, I think there's some other pathways that are important, because if you do these studies in knockout or loss of function mutants of DAF16, you don't see a complete rescue. So there are other pathways that are involved and mediate this effect. Treatment with DDW recovered DAF16. It also increased the levels of manganese superoxide dismutase, which is downstream of DAF16. And the manganese increase in AKT, total AKT, was also blocked in the presence of DDW. And that would obviously cause more of the DAF16 to be phosphorylated. And as I said, the phosphorylated DAF16 actually stays outside of the nucleus. It remains in the cytosol. So that would decrease the translation of the genes that we're discussing. The use of DAF16 mutants demonstrated that other pathways, most likely the NRF2 pathway, and I'll show you one slide on that, might be involved, and not related to DDW, but we think the SED3 is directly involved in manganese-induced toxicity, because if you use a background with a SED3 knockout, you don't see the effects of manganese at all. So I included actually just one slide on the NRF2 pathway. NRF2 is actually probably the most important pathway in regulating oxidative stress. It's sort of the master switch for regulating antioxidant responses. And the way that it works, and we'll just focus on the top part of this slide, is that under regular condition, constitutive expression of NRF2 is primarily in the cytosol, where it's bound to a chaperone protein which is known as KEAP1, KEAP1. When you get electrophiles or increase in reactive oxygen species, a couple of the cysteines on KEAP1 get oxidated, and what that does, it dissociates the KEAP from the NRF2, and then NRF2 can move into the nucleus. It binds to an ARE, or antioxidant response element, and you get upregulation of different phase 2 enzymes, glutathione, hemoxygenase, and probably manganese superoxide and other enzymes and proteins that are involved in battling oxidative stress. I think this pathway might be important and probably should be looked at, because, as I said, it's a master switch. It's elevated pretty much in every pathological condition, and it wouldn't surprise me if, looking at some of the SKN knockouts, which is the homologue of NRF2 in C. elegans, one would see also significant differences in terms of the expression and this pathway as a whole. So I think the NRF2 is going to be important. I think what this model also allows one to do is to look specifically as to whether one can reverse the neurodegenerative effects of manganese on dopaminergic innervation. I've shown you some slides where we can completely obliterate the green fluorescent protein in the dopaminergic neurons. And I think in terms of sort of opening a new can of worms, it would be interesting to see whether one might be able to reverse the effect of manganese on the dopaminergic neurodegeneration with DDW, which should be fairly easy to do. And as far as acknowledgements, as I said, I've worked with Gabor and Ildiko for a number of years now. I'd like to especially thank Diana, who was here, as I mentioned a couple of years ago, who did all this work. And again, thank you for inviting me to be here. It's a pleasure, and I'm happy to answer questions you might have.
We have time. Are there any questions?
It was either too clear or very confusing. Clear. Okay. There's one. Okay, two questions.
Is there any relationship between the cancer genes which participate in cancer and participate in aging?
Well, yes, absolutely. Yes, FOXO2 said they're very important in both in aging. There are many parallels between aging and cancer. Yes, in humans as well. Yes, I mean I'm not an expert on human cancers by any means, but apoptotic cell death is obviously very important, or lack thereof is important in cancer. I think Richard had one.
Because I'm here to learn, I ask silly questions. What's the effect of other metals? Is this manganese effect very specific to, and what's the effect particularly of heavy metals, the toxic heavy metals? Have you looked at that at all? Is there any protection against that? Other toxicities?
Yeah, they're very different actually. The one that's probably most specific to dopaminergic neuron is the manganese. There have been some studies, we've done some studies with mercury. Dopaminergic systems seem to be affected, but to a lesser extent compared to the manganese. Mercury, for example, affects much more glutamatergic system, GABAergic systems. And it's actually very easy to look at those because, as I said, we have GFP labeled strains for each of these neurotransmitters. So what you find actually is that the effects are very similar to what you find in humans. So it actually, at this point, because there has been so much work in terms of the neurotoxicology of different metals in C. elegans, we're probably more so at the stage of defining how useful this model might be for humans. But pretty much you can predict based on the C. elegans what you'll see in mammalian systems. There's been quite a bit of work with cadmium and C. elegans, which does not affect any of these neurotransmitters. What you see is primarily changes in metallothionein, which is also known in humans. Jonathan Friedman has done quite a bit of work on that. There's quite a bit on copper, arsenic, and it's specific to each of these metals. So it's not a sort of a non-specific effect where you might see dopaminergic neurodegeneration.
One more comment only. Thank you for your talk. I only want to say that the DAP16 shows 70% homology with the FOXO. And the FOXO has a very important role in human in the signal transduction and in the transcription factor.
Thank you. Okay, our next speaker is my co-chair, Dr. Olgun, who comes to us from Antalya now in Turkey. And I don't have the title of your talk. Here it is. Deuterination and ATP synthase, stochastic mechanism of aging. So I guess we're going to continue in the same direction.